Alcediag is a subsidiary of a French group named LLC, who recently moved to life sciences. Alcediag developed new tools for personalized medicine, new solution for medical diagnosis with a first focus on psychiatric disorders. The tests are based on proprietary technologies I will come on later on, and we also develop and looking for biomarkers to improve diagnosis and management of the patient. Psychiatric disorders is a major healthcare burden. It concerns millions of people. They are very frequent, most disabling and highly costly for the community. It's really an unmet medical need because of the lack of objective tools. This leads to an uncertain diagnosis and a delay in the management of treatment. And because of the poor efficacy of treatment, there is a lot of drug adverse effect, and suicide is one of them. Preventing suicide is a global imperative as it was noted by WHO in a report last year, and also by FDA in its last guidance. Some treatment has been withdrawn from the market because of their adverse effect in psychiatry. For example, do we have a pointer? Doesn't work. Okay. So it doesn't work. So Paxil from GSK, which has to pay $100 of million to patients. Acomplia, which was an anti-obesity compound from Sanofi Aventis, which was withdrawn from the market 4 years ago. Taranaban, whose development was halted also 4 years ago. Nowadays, the only way to assess any suicidality is to use animal models. They are poorly predictive and raise ethical concern. So EDITOX, a test developed by Alcediag, is a toxicogenomics solution to assess drug-induced suicidality. So the mechanism on which EDITOX is based is RNA editing. So RNA editing is a post-transcriptional mechanism which in fact lead to different variants, to the synthesis of different variants of proteins, and at the end of the day modifies the neurotransmitter function. It needs some specific enzyme named ADAR to be performed. This mechanism is modified by pathology and pharmacology. And a lot, or not a lot, but several receptors has been involved, like serotonin 2C receptor, AMPA, ion channel, with impact in different indications like psychiatry, cancer, cognitive deficits, or cardiovascular, but also autoimmune disease, lupus. 5-HT2C receptor, serotonin 2C receptor has been also involved in drug-induced depression and suicide. And a lot of compounds have FDA alert for that. So today we will focus on serotonin 2C receptor. So serotonergic system has been linked for a long time to psychiatric disorders, and in the end of the 90s, 5-HT2C receptor RNA has been described to be subject to RNA editing. The alteration of 5-HT2C RNA editing has been also described in the prefrontal cortex in some psychiatric disorders and suicide. But also, neuropsychiatric disorders have been linked to immune system activation, like neuroinflammation, and interferon treatment in patients suffering from hepatitis C, for example, or cancer, induce severe psychiatric side effects. All that has been extensively reported, and you have here some of the main papers describing that. So, to have a better understanding of RNA editing regulation, you can see here that if I take the example of a 5-HT2C receptor, there is 5 editing sites that you can see here, A, B, C, D, E, where an A adenosine can be changed into an I, inosine. So this change leads to the synthesis of different isoforms of the receptors. And this happens at the second intracellular loop of the receptor, where the binding with the G protein occurs. So at the end of the day, there is, because of this editing, there is the synthesis of, in this case, 24 variants of the receptor, which differ by the coupling ability. Some are highly coupled, others are badly coupled, and you have all the case. In pathology or in some pharmacological case, there is a modification of the variants which are synthesized. And for example, the variant can be very badly coupled. So the signal transmission is highly perturbed. So to give you an example, if the serotonin is in the synaptic cleft, it will bind to the variant of the receptor. But nothing will happen because those receptors are not coupled. So the signal transmission is blocked here. So this level of regulation is interesting to study, and Alcediac benefit from more than 10 years experience in RNA editing research. We have identified a specific signature in the brain of suicide patient. Of course, it's post-mortem brain. Alcediac has identified an at-risk profile for drug tested on a specific cell line. This at-risk drug profile is similar to the profile found in the brain of suicide patients. And we have also identified specific biomarkers in the blood of depressed suicide patients. I will come back on all these points. So the tool that Alcediac developed for personalized medicine has been concentrated on a specific platform which measures, using a cell test, the RNA editing profile of drugs. The test is named EditTox. And from the blood test, the RNA editing profile of the patient, the test is named EditDiab. So let's go to EditTox. The cell model uses SHSY5Y human neuroblastoma cells, which express the 5-HT2C receptors and editing enzyme. These cells correspond to interferon alpha treatment, and interferon alpha treatment is known and has been described some years ago as an inducer of editing enzyme, and as you perhaps know, triggers depression and suicide in patients. And the profile obtained with interferons that you can see here at different concentration is similar to the profile found in the postmortem brain of suicide patients. So we are able, using new generation sequencing, to analyze the data coming from cell culture or blood, and using a bioinformatic pipeline, we are able to analyze the quality controls to align the RNA sequencing and look at the RNA editing sites. And after that, using a biostatistic pipeline to finally assess from the risk profile of a drug or of a patient. To explain you and give you some example of the validation, so now we have tested thousands of compounds, but I will today explain this study, which concerns 54 market molecules of different therapeutic and chemical classes, which were double-blind tested. The editing profiles were generated, 30 compounds were with FDA alert for depression, suicide, and 24 without. And we have developed an algorithm to assess the neurotoxicity risk of the compound. So, as you can see here, the compounds belong to different classes: antidepressant, antipsychotics, anti-epileptic, mood stabilizing agent, antiviral, anorectic, and others. And to give you an example here, you can see that the black curve is the interferon curve, and some compound like ketonazole, which is an antifungal, aripiprazole, an antipsychotic, and taranaban, anti-obesity, have a profile which is similar to the interferon profile. And those compounds are with FDA alert for depression and suicide. Other, odensetron, antiemetic, ribavirin, another antiviral, and ketamine, which is today used as the anti-suicide compound, has a completely different profile. And those drugs are without FDA alert. So you can see that neurotoxicity is found in all classes of compounds tested, and also varies with concentration. As an example here, the ketonazole at 0.1 micromolar, 1 and 10, or the taranaban, 0.1, 1 and 10. So, having this profile, we have developed a proprietary algorithm to assess the risk of suicide adverse effect of the compound. And as you can see here on the MROC, we obtain this MROC curve and a high specificity and sensitivity. So, as I told you before, we have now more than 100 compounds studied. There is one report, one paper, which was very interesting last year and published in BMC Pharmacology and Toxicology by UCAR group. They studied during more than 12 years the adverse reaction of 11,000 patients treated with 1,500 drugs. And what I found is that the top 5 medicine with report for psychotoxicity like depression were varenicline, bupropion, paroxetine, isotretinoin, and rimonabant. And for suicide attempt, paroxetine, varenicline, citalopram, fluoxetine, and clozapine. And those drugs were detected at risk by our test and with very at-risk profile. So EDITOX is a tool to assess suicidal behavior adverse effect induced by drug. It's a screening test which is able to predict at-risk compound for psychiatric side effect with high specificity and sensitivity. It could be used as a rule-out test in early clinical stage, or also later on to get more information on the mechanism of action of the compound. It, of course, reduces the risk during drug development and save money. So, if we shift now to the patient side, the suicide risk is something which is, of course, a main issue. And Professor Courtet has involved the genetic and epigenetic factor involved in suicide behavior. And trying to define the vulnerability to suicide behavior, he mentions that this vulnerability is likely mediated by an underlying genetic predisposition interacting with environmental and probable epigenetic factors throughout the lifespan to modify the function of neuronal circuit, thus rendering an individual more likely to engage a suicidal act. LCDIAC developed simple blood tests to objectively assess the suicide risk, and we are able to identify, using the test EDIT-DIAC, at-risk individual for suicidal behavior through the identification of epigenetics biomarker. So we have test in one of our clinical trials 77 depressed patients with and without an history of suicidal behavior. And considering 4 biomarkers, we are able to see that there is a significant difference between patients with an history of suicidal behavior and patients without. And the specificity and sensitivity here also are high. So, as you understood, we have one test which makes us a detox, which makes us able to define the RNA editing profile of the drug using a cell test in vitro. Another test which makes us able to have the RNA editing profile of the patient from a blood test. Using the both together, we are able to promote personalized medicine in psychiatry and be able to adapt the good molecule to the good patient, which is quite new in psychiatry. So the take-home message is that LCDIAC epigenetic platforms allow to identify RNA modification. EDITOX is a cell test to assess suicidal behavior adverse effect induced by drug with high specificity and sensitivity. EDIT-DIAC is a blood test to characterize the pathological state of the patient and its evolution during treatment, be able to say if the patient is responder or not responder to treatment. And used together, these tests allow personalized medicine in psychiatry and, as I told you, delivers a good treatment to the good patient. Thank you.
You mentioned about the study looking at patients with suicidal history versus patients, you could say, who didn't have suicidal tendency. You know, the study that you showed the significance. I missed that. Was it patients versus normal people on treatment?
So, no. No, yes and no. So, in this study, all the patients are arriving at the hospital after suicide behavior.
Right.
Okay. Some of these patients, those patients are under treatment. Some of these patients has made one suicide attempt and others have made 10 or 20. Okay. That means that some patients have a real high history of suicide attempt. Okay. They are compared to other patients which suffer from depression or bipolar disorder and who has never had an history of suicide attempt. Okay. We have another clinical study which will compare these two groups to a third one, which is the control one. So it is not patient but placebo.
So, in the patients who say will have suicidal tendencies, because you looked at them whilst they've been on treatment with obviously the form of tendency, but how do you know when you look at the levels and the RNA editing, whatever the markers you use, but how can you tell that actually they weren't any different to start off with?
How I can see the difference?
How would you know that they were any different when the patient started up prior to actually having treatment?
No, all the patients are treated. Okay?
Yeah.
The two groups are treated. And they are treated more or less with the same compounds.
Right.
Some
okay.
have history of suicide attempt and others have not.
And these
Okay.
biomarkers show the difference.
So, and in this condition, which are, in my opinion, is a more difficult condition, okay, because they are all treated, okay, there is a significant difference between the two populations. Of course, the difference with healthy volunteers and or untreated person will be more easy to show.
And I guess you stratify for all the variables.
Yes,
In the
of course, and the confounding factors and so on. So those results are preliminary results. We are now engaged in a very large clinical trial with hundreds of patients.
One question. So this protocol, we test it only to European people?
It's a very good question. So we have several clinical trials. The first one, so we are based in France, in Montpellier. And Montpellier, in the south of France, has a very well-known center to study suicide. And they have, how can I say that, a specific part of the psychiatry in the hospital for emergency and those kind of people. So those people were, let's say, European people. But another essay we are starting will include people from other continents also, because I'm sure that it will be different.